Microscopy has opened an entire universe that's invisible to the naked eye. From the organelles inside cells to bacterial flagella and viral particles, microscopes allow us to observe the structural and functional intricacies of life. In this video, we'll explore the optical principles behind microscopes, compare major microscopy techniques, and provide a guide to mastering the light microscope for routine lab work. Before we dive into how to use a microscope, let's first understand the key principles that define what we can actually see. Magnification is how much larger an object appears when viewed through a lens system. However, magnification alone doesn't guarantee clarity. Resolution is the ability to distinguish two adjacent structures as separate entities. The theoretical limit of resolution for light microscopes is about 200 nanometers due to the wavelength of visible light. This is why electron microscopy is used for structures at the molecular scale. A good microscope maximizes both magnification and resolution to provide clear, accurate visualizations. Let's take a tour of the compound light microscope. At the very top, we find the ocular lens, or eyepiece, where the viewer looks into the instrument. Just below are the objective lenses, mounted on a rotating turret, which provide the primary magnification, typically 4x, 10x, 40x, and, for the highest detail, 100x, which usually requires immersion oil. These objectives are attached to the revolving nosepiece, allowing the user to switch easily between magnifications. The slide rests on a mechanical stage, which can move smoothly along the X and Y axes for precise positioning and is secured in place by clips. Beneath the stage sits the condenser lens, which focuses light from the source directly onto the specimen, and the iris diaphragm, which fine-tunes the diameter and intensity of that light cone. The light itself usually comes from an LED or halogen source, providing uniform illumination. To bring the image into focus, the coarse focus knob raises or lowers the stage for large adjustments, while the fine focus knob sharpens the view at higher magnifications. Finally, the arm and base provide the structural stability that supports the entire instrument. Knowing these parts is essential, but equally important is learning how to bring them together in practice. Here's how to use a compound light microscope effectively. Center the slide carefully and secure it with the stage clips. Always begin with the lowest objective, usually the 4x lens, to locate your specimen on the slide. Next, bring the image roughly into view using the coarse focus. When the specimen is visible, switch to a higher magnification and refine the clarity using the fine focus knob. To determine the total magnification, simply multiply the eyepiece power by the objective lens in use. For example, a 10x ocular combined with a 40x objective gives a total magnification of 400x. To get the most out of your microscope, it's also important to adopt good habits and avoid common mistakes. Here are important tips to ensure accurate microscopy. Always clean your lenses with lens paper before and after use. Avoid using the coarse focus knob on 40x and 100x objectives to prevent lens damage. Use immersion oil only with the oil immersion lens and clean thoroughly afterward. Label your slides clearly and keep a lab notebook with magnifications and observations. Store your microscope in a dust-free environment with the stage lowered and lenses covered. With the basics in place, let's now explore the main types of microscopy techniques, starting with the most common. Bright field microscopy is the most widely used basic technique in biological labs. It uses transmitted white light to illuminate the sample from below. Because most biological specimens are transparent, they often require stains, like hematoxylin or gram stain, to enhance contrast. While simple to use, its primary limitation is poor visibility of live, unstained cells and limited depth of field at high magnification. When we want to observe living, unstained cells, we need a different approach. Dark field microscopy is another optical technique that enhances contrast without staining. Instead of passing light directly through the specimen, a special condenser blocks central rays and only allows oblique light to reach the slide. As a result, the specimen scatters the light and appears bright against a dark background. This method is particularly useful for observing live, unstained, and delicate specimens, such as spirochetes, thin flagella, or very small plankton. Its main advantage is that it highlights fine structures that might remain invisible in bright field. However, the technique requires precise alignment of the condenser, and images can sometimes appear grainy or suffer from glare. While dark field makes transparent organisms glow, phase contrast takes us one step further. Phase contrast microscopy works by converting tiny phase shifts in light caused by differences in refractive index and thickness within a specimen into changes in brightness that the eye can detect. Normally, unstained and transparent samples produce phase shifts that are invisible under bright field illumination. But with this technique, those subtle shifts are transformed into amplitude differences, allowing structures such as nuclei, vacuoles, and organelles to stand out clearly without the need for staining. Despite its strengths, phase contrast microscopy does have limitations. It is more expensive because of the specialized optics involved, and proper alignment of the light path is critical. The technique often requires stronger illumination, and it can introduce visual artifacts such as halos or shade-off effects. Thick specimens in particular tend to reduce resolution. For even greater specificity, especially at the molecular level, fluorescence microscopy takes the spotlight. Fluorescence microscopy uses high-energy light, often lasers, to excite fluorescent molecules bound to specific cell targets. These fluorophores emit light at longer wavelengths, enabling highly specific visualization of proteins, DNA, or other structures. It's a cornerstone of molecular biology and immunohistochemistry, offering localization of molecules with nanometer precision. Yet, to go beyond the limits of visible light altogether, we need another family of instruments. Electron microscopy overcomes the limits of visible light by replacing photons with beams of electrons. In transmission mode, it produces two-dimensional images of ultrathin sections. This reveals the internal ultrastructure of cells, with resolutions down to 1/10 of a nanometer, perfect for observing membranes, viruses, and large molecular complexes. In scanning mode, the microscope explores the surface of a specimen point by point, building a three-dimensional topographic image with remarkable detail, making it especially useful for studying structures like pollen grains, bacteria, and engineered nanomaterials. Both approaches require a vacuum environment, as well as samples that have been fixed and dehydrated to withstand the conditions inside the chamber. Beyond these advanced techniques, there are also specialized microscopes designed for particular applications. The stereo microscope, or dissecting microscope, is designed for low magnification observation using reflected light rather than transmitted. It provides a three-dimensional view using two optical paths, making it invaluable for dissections, electronics inspection, and materials science. Another configuration commonly found in cell biology labs is the inverted microscope. An inverted microscope is configured with the light source and condenser above the stage, and objectives below. This setup is ideal for observing living cells at the bottom of vessels like culture flasks under more natural conditions. It's widely used in cell biology, microbiology, and applications like live cell imaging and micromanipulation. From bright field to electron beams, each type of microscope opens a different window on the hidden world. Learning how to use them expands our ability to explore, discover, and understand.